I'm just kind of cleaning some stuff up before I start. I don't like seeing a whole bunch of files and folders hanging around. I tend to like to clean things up before I mess around. So let me stuff about get rid of this human stuff and just put it into a directory. So I'm going to play around with this file. It's going to be another bed file. It's going to be similar to the GTFs and the bed files I've worked with before. It's just a little bit different, but ultimately, things are the same, right? I have a chromosome. Unfortunately, I'm working with an animal that has a relatively fragmented genome draft, and so it just means that it's not assembled so well up to the chromosome level. So we have it, we call these scaffolds or contigs that are fragments. It's basically the best sequence assembly that we can make, given the tools that we had to make it. And so we can look at this, and so instead of seeing a chromosome number like chromosome 1, 2, 3, 4, we have these identifiers, and it's just PBB, some number. But either way, a different number effectively means a different contig or a different chromosome. Okay, so I'm just going to be playing around with this, and you have access to this if you want to copy it. I have it hanging around in data. So if you want to grab it, it's just gaga.ors.bed. So if you want to play along, if you want to grab this one as well, just copy it from there. So if you want to copy it to your home directory or wherever you're at, just use your period. CP from there to here, right? Again, all that period just means is here, right? So copy from there to here. And so I took this bed file because I wanted to demonstrate a few concepts or demonstrate using tools. And I'm specifically using a bed file that describes the location of olfactory receptors. And so if we've kind of forgotten about olfactory receptors, these are genes. They're reasonably short. They're about 1,000 bases, usually no less than 300 amino acids, usually no more than 400, and somewhere in between there. But if we're just kind of reminding ourselves about olfactory receptors, and it's a gene, and it's specifically a type of GPCR. It's like a G protein coupled receptor, GPCR. And so what we're looking at here, this would be the GPCR. This would be an olfactory receptor. And so you're going to find these expressed and localizing to like nasal epithelium, right? Kind of like right in your nasal tissues. And so like any good GPCR, it's kind of got a complex pathway. But if we're summarizing a little bit about GPCRs, is that they have a lot of these alpha helices in them. And so they also have a lot of hydrophobic or nonpolar amino acids in them. And so what they do is that they kind of cluster on the cell membrane. And so again, this might be the epithelium of the nasal cavity. And so if an odorant comes in contact with this receptor, that odorant won't cross the membrane or anything. It's just going to interact with this receptor and basically cause a signal transduction cascade, which will ultimately lead to information being interpreted by the brain, right? So they're not part of like the central or peripheral nervous system, but they will send information along that pathway to our brain. So we recognize smells, and those smells are largely understood and interpreted by an olfactory receptor. And that's all we need to know. So ORs are about 1,000 bases, and the thing about ORs is that there are hundreds of them in a genome, right? There are hundreds of copies of these olfactory receptors throughout a genome. And so I've been having an undergrad work with ORs in a turtle, or a whole bunch of turtles. So I'm kind of ripping her off a little bit, really to just demonstrate some properties about these ORs. Again, kind of like learning by doing, right? And so in this bed file, what we're looking at is basically the coordinates of every single OR that I could find in the genome, or what we could annotate in a genome. But it's still kind of the same format. So we have the chromosome or the scaffold, contig, whatever it is, the starting base, the ending base, its orientation relative to the contig, and then a couple of identifiers. So I have a few identifiers in here. Right, so this sequence starts on the third base of a contig. So it probably means I don't have the full gene. So like again, a contig doesn't reflect a chromosome, and so contigs kind of fragment a genome. But nonetheless, it's the best we have to work with. So we do what we can. But anyways, this gene is annotated basically at the very front of the contig, which probably means I'm missing part of this gene. Because I'm probably missing part of this gene, and again, like this should be 1,000 bases, right? But it's only 500, so I'm probably missing like the first half 5' region, right? And so the nice thing about ORs is that they're a single exon gene, so ORs don't have any introns in them, so they're really polite to annotate. They're really polite to annotate. Because of that, they communicate information directly, like environmental information directly to the brain. They're really charismatic protein. Anyways, that being said, I give this T nomenclature. I give this annotation of a T, and it just means it's truncated or fragment. Right? I don't have the full gene, and I'm not going to, because I'm missing I'm missing half of the gene. So I just called it truncated. Another weird thing about ORs is that they can be grouped into families. So in this last column is basically a family annotation. So OR 52 is a little bit different from family 10, is a little bit different from family 11, etc., etc., etc. All right, but nonetheless, with this file, we can demonstrate some very handy tool use with cut, sort, unique. Cut, sort, and unique. Right, these three commands that can be piped together, and we can extract a lot of information out of this kind of massive file. So first, let's kind of see how many ORs are actually in this genome. And so I can just do a WC, because again, each line is its own olfactory receptor annotation. They don't cross lines or anything like that. And so in this file, I have 2,000 lines, which would reflect about 2,000 ORs. But also in this file, I have an F and a P. And so what P means is that this region, this annotated region, is probably too short to encode a fully functioning OR, right? It probably means there's a premature stop codon, and it's going to be less than 1,000 bases or less than 300 amino acids. So I'm calling this a pseudogene. It's there. It's just there's a premature stop codon, or there's a frame shift that puts half of the sequence out of frame, and it's not functional, right? So anytime I see a P, it probably means that it's a pseudogene. So that we have these two pseudogenes right next in the file next to each other. So T is truncated. P is a pseudogene. Gene. And F is functional. From what I could tell, given all my annotation pipelines, this is a functional gene. It's fully intact. It's longer than 300 amino acids. The stop codon is in an appropriate place. Everything looks fine with this sequence as far as we can tell. Doesn't mean it's actually used. It just means it's intact. It's probably functional. But right, so I have these 3 types of annotations in here. So, and tells me what family that belonged to. Anyways. So, what I can find out real quick with these tools, for example, is I can count how many are truncated, functional, and pseudogenes. And I can do this with my cut command. So cut, again, I've mentioned this before, but cut really just is like a way to extract a single column out of a tab delimited or any kind of delimited file. So I'm going to pick my column with -F. And so -F, I will then put in, like my column number. So for example, if I want to get the first column, I will cut F1 on this GAGA file. And I'm not going to let it go all the way. I'm just going to pipe it to head so we only see the first lines. But right, so I'm only extracting and printing this first column to the screen. And so I can pick any column I want to, as long as it's between 1, 2, 3, 4, 5, and 6, right? And furthermore, this is a little bit more tricky, but nonetheless, I can also comma delimit this and extract multiple columns. So if I say I want just the first, second, and third columns, I can extract those as well, right? So it's just going to be the chromosome start, stop, and ignore everything else, right? And I can pick whatever column I want to actually print, as long as my column numbers are comma delimited. But that's not necessarily the column I want. If I'm counting how many are functional, pseudogenes, and truncated, I would cut out the 1, 2, 3, 4, and 5. So I'm going to cut on the 5th column on this GAGA. And again, I'm just going to head it, right? And so it just gives us that column information. All right, and so now I'm not going to head this. I'm going to then use a sort command. I'm just going to let it go so we can see how this sort command works. I mean, it's just a sorting. So anytime we see a T, we're just going to find all the T's, put them together. There's probably be an alphabetical order, so it'll probably be F, P, then T. But I'm just going to let this go and let it print out, right? And so right now all my annotations for functional, pseudogene, and truncated have been extracted and sorted, right? And so now let's say I want to count, right? If I want to count how many are here, like of each type, functional, truncated, pseudogene, just counting each of them. Then I can use my unique. Unique. And so recognize by default, if I just run unique, what's going to happen is it's just going to give me the 3 letters, right? These are the only 3 unique things that were in this sorted list. So if I do try to unique before sorting, I'm going to get a little bit of garbage, right? So if I less this, right, it's in the same way, right? Right, it's not very helpful. So if I want to use unique, I have to sort it first, and then I can use the unique command to get what the unique things in that column, right? Basically find all the unique things in that column. Nick, did you get on? I'm assuming you got on by this point. I just saw your email, but did you get on? I'm assuming you're here. All right. Let's see. Okay. So I want to count. I can take this another step further and count. Right? How many instead of using this unique and just printing out the unique things, I can count out how many times, a T was seen or a P was seen or, an F was seen. with my -C. And so it's kind of similar to grep-C, where I can count how many times an instance is present. Right, kind of the same idea. So if I do that, now it's going to tell me that I have 455 functional annotations, 1,200 were pseudogenes, and 356 were truncated. Right, and so y'all don't know this, I know this, but that's a very high pseudogene count. That's really high. To see more than half of all annotations be pseudogenes, that's high. And this is on a turtle. I can show you what turtle this is from. Gopherus gazi. Let's get an image of this tortoise. So this is a tortoise. This is a tortoise, and it's going to be Mojave desert tortoise, but it's it's a cute little turtle, right? Cute little turtle. I shouldn't call it a turtle; it's a tortoise. But yeah, so you're going to find this thing in the Mojave desert. Its genome is available. Honestly, among all the reptiles, I think the turtles are the most sequenced. They're the most represented. So that's kind of why I went on a query spree, because I want to know how many ORs are in each of these turtles. And so this is a tortoise. It's living by sight, right? So most turtles are aquatic, or at least semi-aquatic. In which case, they depend heavily on detecting water-based odorants to find food, mates, etc. But this guy's on land, and quite frankly, I think he's just lost a lot of these ORs. And so what's kind of crazy about turtle genomes is that they're really slowly evolving, and junk stays forever, right? These what I would call a pseudogene would be junk. And so sometimes junk can be cleared out on usually like really fast, really large population, fast reproducing organisms. Junk gets cleared out pretty fast. But turtles hang on to their junk for a pretty long time. And so we can see that in that you would expect, if this were a nice, clean, pretty genome, if everything all waste was gotten rid of as fast as it could, we'd only see 455 functional genes in this. But they're not. Sorry, man. Emailing me and it gives me a buzz, and I hate hearing a ding when I'm talking. Anyways, I lose my train of thought. All right, so these pseudogenes have been hung on to for a long time, and I also kind of know that because like I've done this, we've done this. Me and my lab have done this for like 18 different species of all varying niches, right? Aquatic, semi-aquatic, marine, terrestrial, etc. And they all seem to top out at 2,000. But what changes is the number of functional genes. That seems to be what changes the most. It's not that, and so it's more like these functional things, what would be functional in one species, have been pseudogenized in another, and these pseudogenes haven't been purged from the genome yet. That seems to be the trend among the turtle genomes. Anyways, so that's one thing we can do is count how many are truncated, functional, and pseudogenes, right? And another thing we can count is the number of OR families that are present, and how many of each are present. So let me head that real quick. And so I have, let's see, 1, 2, 3, 4, 5, 6 columns. So I'm going to cut out the 6th column and basically do the same thing. I'm just going to count how many ORs, how many times an OR family was present. So I'm going to cut on the 6th column, just make sure that works. Yeah. And recognize, like, I only call like the file name in the first command, right? Whenever I pipe this, I don't call that file name again. I only call the file name on the first command. -F6, and then sort. Got to sort those out, and then unique -C. And so that's going to tell me the most abundant OR families. OR10 was really common. 11 is super common. 14 is pretty common. 5 is kind of common. 52 is kind of common. Yeah. And so this isn't super meaningful unless we compare it to other turtles. And then we might be able to make some inferences in that regard. But remember, half of these genes anyways, more than half of these genes have been pseudogenized, right? More than half, way more than half, right? So these aren't necessarily the number of functional genes. It's just the number of genes total. But it's possible I might be able to, let's just see what happens. I'm curious, I don't know what's going to happen. Let's just say I cut out the 5th and 6th column. And so that's going to be pseudogene and OR family and do the same thing. Just sort them and unique and see what happens. I don't know what's about to happen. Can't read it. Not easy to read. But of all of our functional, at least tells us of functional genes and where they're found at. And so among the functional genes, it looks like OR11 and 52 are the most common. Yeah, I don't see anything higher than that. 11 and 52. Pseudogenes. Let's look at pseudogenes. There's all the pseudogenes. And most of the time when we look at ORs, there's usually a linear relationship between the number of functional genes and pseudogenes. So it's not always surprising. You won't find like a whole lot of one family has been pseudogenized. You just see this linear relationship. Yeah, but OR10, a lot of 10s have been pseudogenized. A lot of 14 have been pseudogenized. What else? 50, yeah, 52, kind of frequent. And who cares about the truncated, because the truncateds is the most kind of garbage. I can't even tell what it is. I don't know if it's pseudo. I don't know if it's a pseudogene. I don't know if it's functional. I just know it's truncated. So those are kind of the most garbage. And quite frankly, given how many contigs there are in this, seeing only like about less than a quarter are truncated. So in all reality, it's not that bad. I've definitely seen some genomes where it's like the truncated outnumber the number of pseudogenes and functional genes. Anyway, all right, and so that's kind of like using cut, sort, unique to again just to extract some information and learn some stuff about a file. Not worrying necessarily how I made this file, but reading the output, analyzing the output of this file. But we can do one more thing. And so this is a weird thing that ORs do. It's not really weird, but it's kind of an awesome thing that ORs do. Is that ORs, you usually find them neighboring other ORs and nothing else, right? So you'll see just this whole block of a genome just dedicated to OR genes. And so they multiply pretty frequently through like a tandem duplication mechanism, right? So you'll see like a hiccup in DNA replication, and so the same gene will be copied twice, and they'll be right next to each other. You'll see the same copy of a gene right next to each other, and so it's called a tandem duplication. And we can study this tandem duplication trend with these ORs. Now, this genome's not going to be the best one to do this because it's not very contiguous, right? It's the contigs, the scaffolds are fragmented, so it's not going to be perfect. But let's see. But we can use these modules in bedtools. So we're switching from bedtool or cut, sort, unique to bedtools. And again, bedtools really help analyze bed files. And one thing that we can do, so we've seen interact, we have seen intersect bed, which is a module of bedtools. Let's just get out that whole bedtools menu of things that we can mess with, right? So these are all of the subcommands that we can run from bedtools. And what we can use is cluster and sort. So sorting, right, like any good sorting algorithm, it's just going to sort everything from first contig to last contig, first base to the last base for every contig, just in case something's out of order in all of this mess. And cluster, with cluster we can find overlapping or almost overlapping regions. We can annotate genes that are overlapping or almost overlapping. So we can do that real quick. So as opposed to doing like bedtools, sort, bedtools, cluster, we can do sort, bed, and cluster, bed. Right, so both sort bed and cluster bed, you can just type out cluster and tab over. It's going to be the same command, right? That's the same thing as running bedtools cluster, right? Those two commands are interchangeable. Anyways, so, but we can't cluster without sorting, right? Just like we can't count uniques before sorting. So we need to make sure that this file is sorted. There's no guarantees that it is. So I'm going to sort bed, and so with sort bed I have to call an I. I have to use my this I option to read in this bed file. So instead of doing like a WA or an A or a B, it's going to be an I. Sort bed -I, and then the input file, and so that's going to be the GAGA. Okay, and so that's just going to sort everything out, make sure everything's in the right orientation. We start on the first contig, we start with the first base of that contig, and sort everything out on this contig. So right, so that's all. There's only 2 genes on that contig, 4 genes on that contig, 2 on that one, and then a whole bunch on this contig. 1043. Has a lot of ORs on it, right? So that's kind of unique in itself. But with cluster, whenever we run cluster bed, right, all this stuff can be piped. Sort bed can be piped to cluster bed, and I don't have to call anything with cluster bed, but I will show the options of cluster bed. All right, and so the only 2 options that we have to care about is a -I option if we were running this by itself, but we're not because we're piping it in, and this D option. And so what this D does is set a feature distance, right? So instead of finding like overlapping genes, I can ask cluster bed to find genes that are within, say, 1,000 bases of each other, or 10,000 bases, or 100,000, etc. So let me sort that real quick, sort bed, and pipe it to cluster bed. And so I got to use my D option. Otherwise, it's just going to try to find overlapping genes that overlap by 0 or less base pairs, right? That's not going to be realistic for this kind of gene. For TEs, that's super realistic, but for a gene, that's not very realistic. So I'm going to set a distance of just like 10,000 bases. So basically, I'm going to try to find all the genes that are within 10,000 bases of each other and give me a cluster number for those genes. So let me just pipe this to left so I have a little bit more control over it. Okay. Okay, and so now we have this new column. When we use cluster bed, we have this new column. And so this column is a cluster ID column, right? So this is the first cluster found. It's on this 30th scaffold, and it includes these 2 pseudogenes of OR5. Almost all 4 of these ORs on 60, on the 60th contig, we see all of them are OR5, and they're all pseudogenes, but they're all within 10,000 bases of each other, right? So this whole area, this whole neighborhood is about, oh gosh, I can't math. It's about 50,000. Yeah, it's about 50,000 bases. right? We start at 550K and go to almost 600K, right? So that's a pretty long neighborhood. And so there's a decent distance between these genes, but whatever. And that didn't work. All of this stuff, I bet it forms a cluster. I'm going to change from 10,000 to 100,000 because, quite frankly, when we're talking about distances of hundreds of millions of base pairs, 100,000 really isn't that long. So let's just look at clusters, genes that are within 100,000 bases of each other. Okay. It's just reclustering them, right? It's just reclustering. Okay. And so, yeah, so now everything on this 1043 scaffold seems to be clustered. Like every one of these ORs is within 100,000 bases from another OR. And most of them are pseudogenes, right? That's no surprise there. Within the whole genome, most of these things are pseudogenes. And we see like some of them are in the reverse orientation, some of them are in the forward orientation. And we generally see a mix of OR types, but still 4 and 14 are pretty common. 11 is pretty common too, and you have a whole bunch of 11s right next to each other. That's kind of neat. I think ORs do crazy things, and I think it's pretty awesome what they do. Anyways. So, well, let's just see how many clusters that we actually will end up with. So I'm going to tail this just because it's going to give me the last 10 lines and tell me how many the last cluster number. And so the last cluster number is 953. And again, $1 says that there are way more clusters. Like the clusters are much bigger than what's being presented here. It's just because the fragment, the genome isn't fully contiguous, right? We don't have a full chromosome level assembly. But that's okay, right? That's a single contig, that's a single contig, that's a single contig, et cetera, et cetera, et cetera, right? So we just see these genes that are just on a single contig. And these are kind of, it's like this whole thing is just a truncated gene, right? This whole thing isn't even that long. This whole contig is basically an OR gene, and it ranges, this whole contig is not going to be more than 300 bases, right? So that's not very helpful. But nonetheless, it does tell us that these ORs aren't dispersed randomly throughout a genome. They cluster. Most of them will cluster. Because we have fewer clusters than genes present. Remember, we have like 2,100 genes in here, but 900 clusters. So at least half of them, it suggests that half of them are found in a neighborhood of 2 genes or more. In most cases, it's going to be a lot more. It's just this is a fragmented genome. Anyways, let's kind of, we can have this file if we want to. I can just let that run and you save it if I wanted to. Like, if I so wanted to, just so I don't have to write these commands out over and over and over again, I can just save that file as needed. Okay. But let's try to find out what the biggest cluster is and where it's at. And we can do that with our between sort and bet. Cut, sort, and unique. Sorry. So I'm going to cut. If I'm going to cut something and I want to know what's the biggest cluster, I'm going to cut on the 7th column. So I'm going to cut this column out and basically count it. Now it's already sorted. So I don't have to resort it. I can just cut it and unique it. But it's going to get a little bit bad. Cut dash F7 for the 7th column. And I'm just going to cluster it. No, not cluster, unique it. Unique. And I'll even do a unique -C. All right, and so we do have kind of a problem, because now it did do what I wanted it to do, right? It does count how many ORs are in each cluster, right? 4 has 34, 12 has 20, 19 has 21, etc., etc., etc. But it didn't sort and tell me what's automatically the biggest. And so I can add one more sort. Let's just do that real quick. Pipe it to another sort command. And I'm going to add a -in option. So it's basically, so sort, let me try 1 to 100, seek 100. Yes, okay, so just to demonstrate what sort does and how computer sorting works, because computer sorting does weird things. It's not the typical sort. So seek 100 basically means print all numbers between 1 and 100. Just print them out. This isn't a command you need to know. I'm just demonstrating this command and demonstrating how sort works. So I'm just printing 100 numbers. And so if I sort them, what's going to happen is that it's going to cluster all numbers that start with 1 first. And so this is just an annoying thing that computers do, is they don't like, if I have 3 digits, it doesn't like to see 1 to 100. If I had 3 digits in a group, computers would sort if it were like 001 or 010 or 100, right? It's going to sort that better because now everything all has 3 digits in it. It starts at 001 and goes up to 100, right? That's how computers like to sort. And if we see this, we'll see that, right, it gives us 1, then 10, then 100, and then 11, 12, 13, 14, 15, 16, 19. Kind of a silly, weird way that computers sort numbers out, right? This is not a necessarily numeric sort. It's just, it's a computer crap sort. But we can fix this. And we can fix this with a -in option. And so with -in, now it's going to sort it numerically and do what it should, right? So now 1, 2, 3, 4, 5, 6, 7, 8, 9 to 100. Okay, so I kind of have to do the same thing here. If I want to cut out the 7th column, unique -C, and then sort on that number. So I want to sort on this first number, right? And give me the largest number last. So I can do a sort -in. And so now it's going to do numeric sort, and it's going to give me, it's going to sort them out from the smallest clusters to the biggest. And so now everything's sorted out, and we can see that the biggest cluster has 34 genes in it, and it's at that 4th cluster that we've seen before. The next biggest is 21, 20. But again, I promise, like, this does not necessarily reflect reality, it just reflects the genome draft. Most likely these clusters are, you know, hundreds of genes large. And a lot of these clusters that seem independent, they probably overlap with each other. They probably do form a contiguous single cluster. It's kind of neat stuff. But I'm going to call it there. I think we're 40 minutes in. That's probably enough playing around with GTFs and beds. And whenever we come back from spring break, we'll kind of take a hard left turn and go in a different direction. We'll start talking about annotate, like measuring gene expression, identifying genes that are expressed, annotating genes that are expressed, etc. So we'll move away from phylogenetics and look more at gene expression and how we can measure gene expression. All right. Until then, enjoy your spring break. Try to relax. Good job to all the presenters from yesterday. That's like almost half the class. Half the class, I think, presented. So good job to y'all. And I'll see you after the break.